Chapter 7, Section 6, Regulation of Chemotaxis. Now remember, chemotaxis is basically movement of these cells, these unicellular cells, either towards something that's an attractant or away from something that's a repellent. Good, bad, literally. And it's all due to gradients, gradients that are constantly changing. Okay, as things diffuse through the liquid, diffuse through the air, this gradients are going to be changing. More of something in this area versus that area, which will change over time. Bacteria use, again, a two-component system here. There's going to be some form of a sensor in the cytoplasmic membrane, and then there's going to be proteins inside that will be modified. They'll modify other pre-existing proteins, setting up a little signal cascade that'll cause flagella to move one way or another. You look here, you can see Pseudomonas aeruginosa, the little green dots are bacteria. Here are some epithelial cells. The epithelial cells were damaged. Basically, they burst open. How? Doesn't matter. Intracellular contents were spilled out and starts to diffuse away. You can sit there and watch over time as more and more green dots come from up here towards the damaged cells, because the damaged cells are basically now open bags of food. This is acting as a positive chemoattractant, pulling in, the bacteria as they are swimming via the use of all their flagella. Response to signals here for chemotaxis is going to be based on, like I said 2 slides ago, a cascade of proteins, similar to, but not quite the same as what you've learned about in intracellular molecular and cell biology. These proteins, many of these proteins are what they're referred to as methyl-accepting chemotaxis proteins, MCPs. These are sensory proteins that sense attractants, repellents, depending upon which MCP it is, and then will activate some proteins in the cytoplasm. You'll see that these will cluster together in the membranes, forming what are referred to as chemoreceptors, chemical receptors. There are going to be some of these chemoreceptors that are responding to attractants, sugars, things like that, and then things that are a repellent, changes in pH. What we do see is that one of the sensor kinases, Che, is an autophosphorylator. Okay, autophosphorylates, self-phosphorylates, doesn't need some other kinase to phosphorylate itself. It binds to its signal, whatever its chemical signal is, and changes its conformational shape, which means it can now strip off the terminal third phosphate from ATP and phosphorylate itself. It will then transfer that inorganic phosphate to its target, Che, which is the regulator that goes and starts the process of flagellar movement. You see here outer membrane, cytoplasmic membrane, and this array of receptors and the cytoplasmic receptor array, so that things that are coming into contact with the cell are going to hit these arrays. And what you don't see here, because they're too small to see, are the regulatory proteins, the ones that are going to be activated and cause things to happen. All right, so when we're looking at this, we're going to split this cell down the middle here, okay? This is what happens for an attractant. This is what happens for binding. I'm sorry, a repellent. MCPs, okay, that are in these arrays, these clusters, gets bound by an attractant. Remember, it autophosphorylates itself. It autophosphorylates itself, then phosphorylates a couple of others, JW, JA. Signal process, boom. Flagella starts spinning. But here's the thing. The flagella starts spinning in a counterclockwise fashion. By spinning counterclockwise, this is going to give it a run. A nice run, almost a straight run towards something. Now, it's not always going to be straight at the source of whatever that attractant is, because you got to remember this is random. But given enough starts and stops, it will get ultimately to where it needs to be. High concentration of whatever that attractant is. The repellent, autophosphorylation, JW, JY. JY will then go and activate proteins associated with the flagella. But here it's causing a clockwise. This started a counterclockwise rotation. This starts a clockwise rotation. This clockwise rotation, you would think would be the same, just because things are going one way versus another, that the way the flagella acts, the way the cell acts would be the same, but no. This is more of a tumbling activity, more of a random run, stop, run, stop, repellent, attractant. So JY ultimately governs direction. Counterclockwise, the run, nice and smooth. Clockwise, the tumble, it's random. Random movement, random, MCPs bind the repellent, release attractant, JY becomes phosphorylated, interacts with flagella, causing a clockwise tumble. MCPs bind attractant, JY doesn't get activated, doesn't interact. Other proteins, other things will be triggered at the same time, causing a counterclockwise or a run, a nice smooth. But as in all signal processes, you need to be able to reset. So there's going to be some form of a feedback loop. Remember from that figure, JB. JB goes and resets everything. It will go through and demethylate, dephosphorylate the signal proteins that are associated with Che, that operon, resetting the system. We see that this similar processes are coming into play for other taxis, phototaxis, aerotaxis, responding to light. So there are sensors that will be triggered due to photons of energy. There are others that are going to be triggered due to oxygen, high oxygen, low oxygen, and whatnot.